Welcome back. Let's do a quick review of meiosis and what's happening in meiosis. So remember, we need another process of meiosis because our cells in our body are diploid or 2N. And if we would take those cells that were diploid and they would fertilize each other, then the fertilized cells would have too many chromosomes. So in the process of meiosis, our gametes become haploid. So when they are fertilized, they will become diploid. So using these haploid cells after the process of meiosis will also produce variation, because during the process of meiosis, we are going to have something called crossing over and something else called independent assortment occur, both of which produce variation. And then the act of fertilization itself is random, and that is the third item that produces variation. So let's get started with reviewing meiosis. So in its most simplistic form, whenever you see these pictures with these red and blue chromosomes, this is the most simplistic form of meiosis, leaving out all the details. And what we're going to have is one duplication, as you see here, going from step 1 to step 2, followed by 2 divisions. The first division going from step 2 to step 3 goes from 1 cell to 2 cell. And that's where the cell is going from diploid or 2N to haploid. This is the step which makes it haploid. The second division is going to be called meiosis 2, where we are going to split up the sister chromatids and end up going from 2 cells to 4 cells. It is staying haploid at that point. So let's get started. Before meiosis, during interphase, the homologous chromosomes. So we have a chromosome from mom and a chromosome from dad. Those are called homologous chromosomes. While in interphase, they're going to duplicate, and you will have sister chromatids of each being formed. All right, so now in meiosis, in the first step of meiosis, so for meiosis 1, during the first phase, so during prophase 1, in the beginning, we have, before this, we have mom's chromosome and dad's chromosome. That's each are duplicating. So now we have this homologous pair of chromosomes, each of which now is sister chromatids. All right. So in meiosis 1, now if the 4 of them would be next together, the 2 pairs of sister chromosomes, they would form a tetrad. And in meiosis 1, they're going to line up as a tetrad, and they're going to separate. But we are still going to have the sister chromatids at the end of meiosis 1. So the first division takes us from 2N to N, in other words, from diploid to haploid. In the first division, meiosis 1, we will have exchange of genetic material during crossing over, and we will have random alignment during the process of independent assortment. So the exchange of genetic material happens when the long arms of the homologous chromosomes basically will break off, and they will exchange their genetic material. The net result is you end up with recombinant chromatids for 2 of the chromatids, and then one of the paternal chromatids is exactly the same as before, and one of the maternal chromatids is exactly the same as before. So this step is known as crossing over. It allows for exchange of genes between homologous chromosomes. You end up with 2 recombinant chromatids, 1 chromatid, which is like the original maternal chromatid, and one that's like the original paternal chromatid. And this occurs in prophase 1. Then these sister chromatids from the dad and sister chromatids from the mom will then go into metaphase 1, where random alignment will occur. And so what happens is the mom's chromatids and the dad's chromatids line up in the middle. And each mom and dad gets to decide, do I want mom on this side and dad on this side, or do I want mom on this side and dad on this side? And that happens for all 23 pairs of chromosomes. So this is just showing, for instance, 2 chromosomes, and one of them has the 2 moms on one side and the 2 dads on the other side. And so if they would divide up, you would end up with 2 cells, both of which had all mom stuff, and 2 cells, both of which had all dad stuff. Whereas if they alternated, you would have 2 cells that would have both of which would have moms for this chromosome and dads for that chromosome, and the other 2 cells would have moms for this one and dads for that one. But remember, this is occurring in metaphase, which was after prophase. Okay, so this is after the crossing over. So this simplistic diagram isn't accurate. It would look more like this. And so you would have to show evidence of crossing over. And so when these cells separate into their 4 cells, you would have, for instance, if all of dads are on one side and moms on one side, you would have 1 cell that shows all dads original, 1 cell showing dads with the recombination, 1 cell showing moms with the recombination, and 1 cell showing moms original. Whereas this cell with only 2 chromosomes where they were not lining up moms on one side and dads on the other side, you would have 1 cell with the short mom's original and the big dad's original. And then 2 cells, both showing recombination, one with dad's big recombinant and mom's small recombinant, the other one with the opposite, and then the 4th one would be with mom's big original and dad's little original. So this process is known as independent assortment. There's a randomness to the lining up of the homologous chromosomes, and it occurs in metaphase 1. So after anaphase 1 and telophase 1 with cytokinesis, we will then have meiosis 2. And in meiosis 2, the sister chromatids will separate and you would end up with 4 cells. And this is how it would look in its most simplistic form. But remember, we would have to have evidence of crossing over. So it would look more like this, where you would see evidence of crossing over. Once again, only showing 1 or 2 chromosomes. You don't see much variation going on here. But meiosis 2 is the 2nd division. Remember, we start these cells haploid and they end haploid. This looks like regular mitosis, except for the evidence of crossing over that occurred during meiosis 1, during prophase 1. So with 46 chromosomes in humans, because of the independent assortment that occurs in both meiosis 1 and meiosis 2, it's possible to have over 8 million different combinations. And that is why you don't look like your siblings. So let's take a brief moment to review the net results of what happens with mitosis versus meiosis. In mitosis, we start with a cell that is diploid. We have cells that are diploid during the process, and we end up with cells that are diploid at the end. We end up with 2 daughter cells, both of which are identical to each other, and both of which are identical to the parent cell. In meiosis, we start with the cell that is diploid, and then in the first meiotic division, we end up with a cell that is haploid, and in the second meiotic division, we still end up with a cell that is haploid. We end up with 4 daughter cells, none of which look like each other, and none of which look like the parents, and the 4 daughter cells will show evidence of crossing over. So Hopefully you understood all of this. If not, please go back and look at my earlier sections where I have a much more in-depth review of mitosis and meiosis and go through that more detailed description of what is happening. Otherwise, go here and take this picture and review all the steps of meiosis 1 and 2, naming the stages, the specific events, what's happening, and make sure you can identify what is haploid and what is diploid. And I will see you soon when we come back and we actually start looking at male reproductive structures. Thank you.